次のスピーカーは水メリットで
Thank you so much for inviting me to speak today and to be with you. 2 years ago, we had a wonderful dinner together in Dublin, and last year with many of you in Boston. That's a picture, and I'm really very honored to have this new collaboration with many of you and to be here in South Korea. So Dr. Kim and I spent the last couple of days together, but we didn't compare our slides. But I think you will see by the time I'm finished talking that we're very aligned in our thoughts about the future for ALS trials. So I think we've had tremendous progress in the last 25 years for people with ALS. And I think most importantly, we do have, in a way, 4 approved drugs in some condition throughout the world for people with ALS. We obviously have a lot more to do, but we've had some significant progress. Riluzole being the first one in 1995, and then Dexta in 2011, which is approved in some countries, not in all, as a symptomatic treatment for people with pseudo-lobar affect. And more importantly, a recent small study led by Dr. Richard Smith suggests it might also help bulbar function, including swallowing and speaking. And here you all know very well the conditional approval of Neuronata R as a stem cell treatment for people with ALS, and I'm looking very forward to the talk later today to share more information about that. And more recently, the approval in 2015 in Japan and South Korea and other countries, in 2017 in the United States of Radicava. But along the way, there have really been a lot of things that have led to this success. And as Dr. Kim mentioned, really the beginning was identifying the first gene and giving a clue of what could go wrong to form motor neuron disease, and then to allow the ability to create some models for the disease. And now in 2018, many of the genes, and we have many models, and there's so many more people studying the illness. Throughout this timeline, there's been creation of networks. And our NEALS network, which I'll tell you a little bit about, and in particular why I think forming something like PACTALS can really accelerate treatment development. I'm so excited about that happening here that you started in 2014. NCALS formed in 2012. And along the way, people have really developed the tools that we need to accelerate how to develop treatments for ALS. I mean, ALS is really a pioneer for CNS drug discovery. ALS was the first illness that any antisense oligonucleotide approach or gene therapy approach was used, and that was in 2011 for SOD1. Now, since that time, there's now an approved drug for a childhood form of motor neuron disease, SMA. But it was really the ALS study that launched the first steps of how do you do this in people. And now there's an active phase 1/2 study in people with SOD1 ALS using this approach. And there's other gene therapy approaches coming. And most importantly, there's really a phase 3 trial being planned, a global trial for this antisense oligonucleotide treatment for SOD1. And hopefully that's a study that PACTALS can have a huge impact in. Also, we're really pioneers in how do you deliver treatments in the spinal fluid, how do you deliver in the spinal cord, and how in general do you do clinical trials. So I'm going to highlight a few of the areas where I think a consortium like PACTALS can make a big impact. When our patients see us, they ask us, why, if you've known about the illness, usually they say since Lou Gehrig, but since Charcot, how come we don't have treatments? And as you know, it's an enormously complex illness that involves many, many different pathways. And as we heard from Dr. Kim, it might be that there's different pathways in different people. But this list is actually also, I think, hope for our patients, because there are several targets here where there are treatments that are either on the shelf or ready to go to people, and where we can start to move them into patients today while we develop even better ones for the future. So I'd like to keep a running list of what's in clinical trials and what's coming in clinical trials, and there are a lot of things going on globally. So the things in dark blue are the 4 drugs that are in some form approved, the Riluzole, Derivaone, Dexa, and the Neuronata. In light blue are drugs that have had some positive phase 2 data in people. So that gives hope. Now we know that not everything that's positive in phase 2 turns out positive in phase 3. I think the ALS field has learned that in the hard way, but it still gives hope. And then everything with an asterisk are trials that are currently enrolling people in some country. And this list also could be missing some. It's actually a challenge now for patients to find out all the studies that are going on in the world. And so there's a couple websites. Our NEOS consortium keeps a running list of trials and tries to help patients find a trial near them. We also have a 1-800 number to help patients find trials. And I wanted to highlight that because that's something that an organization like PACTALS could make a huge impact to help patients be part of research. We also have an email for that. And then, of course, there's always clinicaltrials.gov. So this gives a lot of hope and also shows that people are targeting different pathways. Many of the drugs are targeting neuroinflammation. Some are targeting hyperexcitability. Some are stem cell trials. And then a whole bunch of new targeted gene therapy trials. So what are we going to do with this list? So this is a good problem to have as an ALS field. We have a lot of great ideas about targets. We have a lot of potential drugs and many, many investigators. Yet we have the challenges that you heard about by Dr. Kim of how do we pick the right therapy for the right person, and how do we screen these more efficiently. If you have a pipeline of 50 or 60 drugs, if you do them, test them one at a time, we're going to be here 100 years from now. So we do have to learn from our colleagues in oncology and other fields who have developed much more innovative ways to screen drugs effectively in patients, such as platform trials or other approaches. Because the one-on-one, the one at a time, will not work for our patients. So I think that we have a lot of opportunities, but we have challenges of how to do this the right way and in a much more stratified way for our patients. So as you heard from Dr. Kim, one of our biggest challenges is the disease heterogeneity. But I think there's a lot of hope with the tools that are now being developed around the world to be able to, one, image the illness in people, to develop screens through IPS lines or eye neurons, as you heard about, or to use really clinical phenotype to separate the patients. But we also hear loudly from our patients is that they want access to treatments. So sometimes our trials are restrictive for good reason, but that also means that the majority of our patients can't be on the trials. So I think as a community, we also have to think about how do we make sure there is something for everybody and that the access, barriers to access are limited. So in 1995, shortly after the discovery of the first gene, perhaps a little naively, we thought the treatments were around the corner, and we wanted to develop really the tools to figure out how to develop treatments in people with ALS. So we formed a consortium in New England. So it was a small group initially, but this is now a group throughout North America of more than 100 ALS trial sites with more than 500 members. And in this network, it includes physicians, nurses, physician assistants, physical therapists, anyone who touches people with ALS, as well as the patients themselves. And so this consortium's been around for 20 years, but one of the things that we're, I think, most proud of is really developing a cadre of people who know the illness, who have centers near patients, so patients don't have to travel far, and creating shared resources to really accelerate discovery and break down barriers. And those resources include biofluids, DNA, cell lines, as well as shared clinical data. I know we're going to have a series of talks later today from several of you on initiatives similarly of how to really build a network and build repositories together. The other advance that we tried a couple years ago was the idea of a central ethics review for the network. And this was made possible in the United States because of a change in law that allowed hospitals to rely on each other for ethics review. So instead of 70 different hospitals making a decision on ethical review of a protocol, one group could do that, and all the centers could rely on that. And so that initiative alone has shaved off about 9 months of startup for clinical trials. So it's something we now do for all our trials. The other initiative was, with all this big pipeline, we needed more people, more young investigators attracted to ALS and learning how to be a leader of a multi-center or single-center trial. So we put on annual trainings now for young investigators or older investigators to learn how to do that. And we now have more than 80 physician scientists who are ALS physicians and are ready and able to lead multi-center trials. Of those 80, 25 are leading studies right now. And more recently, we really have brought in the patient voice, which is so critical. And now we have over 100 patients and their families who've come to our meetings, have learned about trial development, are on our steering committees for trials, and give us their voice of what they need and how we can continue to do better. But this took some time to build. As you can see, it would look a lot younger in those days in 1995. But we started as a small group of 9 people, really under the leadership of Dr. Ted Munsat, whose vision this was to create this group. And it took meeting monthly and brainstorming about what we wanted to do together, but with a shared mission to try to accelerate treatments for people with ALS. And so right now we are running 27 trials together. Some of those are drug trials, some of them are biomarker or fluid-based or imaging type studies. We have 124 sites in US and Canada. We also have some sites in Israel and South America. It's not really restricted to North America. We have a large biorepository of fluids, and we're very excited that recently the foundations gave out grants to 12 different investigators to use these samples to develop new biofluids. These samples are available to any ALS scientist throughout the world or in academic labs or in pharma. We've really focused on trying to share data. So I'll tell you a little bit more about PROACT, but this is a data set of all the placebo arms of past clinical trials that have been merged together as a free open data source. And so there's over 11,000 people's data in this, and this has been used to develop predictive algorithms for disease course, for companies to have data to model for developing their trials, and for academic groups to study potential prognostic factors. Similarly, Neurobank is expanding that outside of the clinical trials, and that's about capturing data from clinics, from many studies, and really creating a big data system so we can learn about ALS from really every patient that we see. On the right is really our efforts to train the field of people who care and love about ALS. It's very exciting now as a neurologist to see people coming to apply to neurology residency and saying, I want to be an ALS physician. That was pretty much, I think, unheard of 20 years ago, and it really shows the excitement of the science and the potential for people to make a difference. So we focused a lot early on training people. So we trained principal investigators. So we trained our sites to be top sites, to know all the rules of being clinical trialists, and to be ready for when there was a trial available. We also spent a lot of time, and we continue to do that, to train people at the sites on how to do the outcome measures in a reliable, reproducible way, so that, again, when a trial is available, a company or an academic leader could go to the site and the personnel there are already trained and certified to do all the ALS outcome measures. As I mentioned, we're really focusing a lot on patient partnerships, and what we really heard from the patients is they would like us to really make this whole process easier for them. It's hard to come to clinic to be part of studies. It's hard to be there for 8 hours, 10 hours. Can we start to move some of this into the home or use technology? And so that is, I think, a hot area for us all to work on. And can we help people actually know what's going on and know what trials are available and how they can help and be part of the whole effort? And so we have a website where we have monthly webinars and lots of information for patients to help. And more recently, we really wanted to try to accelerate some of the great ideas coming out of people's labs to get into patients. And what we were finding is that while large pharma is very interested in late-stage trials for ALS. There weren't so many originally in the early phase trial. And so we partnered with a couple foundations to fund people who had a drug ready to go to patients in an early phase trial, with the idea that we would lower that barrier, fund those studies, and then if they were positive, that could be brought forth and by a pharma into phase 3. And so, for example, the study I mentioned of Udexta, where we showed that it can improve bulbar function, was the first study funded by this mechanism. And so this mechanism has been present for about 7 years. There's a call every October. It's not limited to the United States, it's any country. And I'm very proud that last year that we had our record number of applications. We had over 20 applications, and these two foundations funded the 3 top proposals. And the hope again is that there will be a call for phase 2 studies this October as well. So this is just a view of our trial finder. And I think this is one of the things we've heard the most feedback from our patients, that how much it helps to have, 1, someone to call, to ask questions about, and 2, a place to look to find what's going on close to them. So I want to go back and talk about the challenge. The challenge you heard from Dr. Kim is that we know clinically, and likely biologically, driven from the genetic data, that this is a very heterogeneous disease. And so this has made our past trials very challenging. And it also means that if we just do small studies, whether they're with controls or without controls, we can be very misled. And I think the field has learned that from several drugs, including, for example, lithium and dexpramipexole, that there's no, at the moment, shortcut. But there is a big need to develop ways to screen drugs in short time period, in a small number of patients, to develop those surrogate markers so that we can eventually not need to do long, large survival studies. What the PROACT database taught us is that the disease is probably not linear in everybody throughout the course of their illness, that over a quarter of the patients that were in those trials had up to 6 months where they did not change at all in the measures that we had, like the ALS-FRS and the breathing. There's 16% who don't change in 12 months, and a smaller amount in 18 months. And that means in these small studies that if by chance you have any of these people in your group, you could be very misled in both ways. You could think your drug doesn't work, or you could think that your drug does work. So it's important for us to really understand this and find better ways to do these early phase studies. The classic example of this really was the dexpramipexole study, which was for Biogen. And this was a drug that had what we thought was a very positive phase 2 study, where it was a randomized placebo-controlled trial, 3 doses versus placebo, but there were only 25 people per group. And but it did look like there was a dose-related slowing of disease progression on the left on the ALS-FRS, and the survival benefit shown on the right. And this is where we developed a new outcome measure called the CAFS. And this is a way to look at ALS-FRS and take into account mortality. And this is a type of a joint rank outcome that the FDA now likes very much for pivotal phase 3 trials. So we went into the phase 3 with a lot of hope, and the trial rolled very fast, over 5 months, over 900 people in a global study, but it was resoundingly negative. This is on the CAFS, but also on the individual components on ALS-FRS and survival. So this was, in a way, a wake-up call, again, to the community that this heterogeneity, or taking all comers in trials and not understanding their underlying biology, can be particularly misleading in small studies. So what are we going to do about it? So the good news is there's lots of activities really globally trying to understand this heterogeneity. And one of the studies that we're working on, which is very similar to the studies that Dr. Kim and others here are working on, and also in Australia, is to try to really understand this. So in Answer ALS, we're enrolling up to 1,000 people with ALS with very good clinical follow-up. They're all giving biofluids, and they're all having their DNA sequenced at the New York Genome Society. But we're also generating these iPS cell lines from every patient. and we're doing full omics on those lines, whether they're pushed to become neurons or glial cells. And then we're really partnering with different big data analytic groups to try to see if in that massive amount of data, can we pick out different cohorts of people that have similar biologies. And then ultimately the goal will be, can you use people's own derived neurons to screen drugs. So this study is well on its way. It's enrolled about almost 900 participants, and many of those lines are made. And the other exciting thing about this work, because it was funded all from foundations, is that all this data and the lines are all, again, publicly available for any ALS investigator. Now, we don't know if 1,000 people is enough, or is it too much. I think we'll learn that as the first set of data come out. But there is actually a backup study already underway that Biogen is supporting through Columbia, where they have another set of 1,000 patients who have the same clinical data being captured with the same system Neurobank. They're not making all those lines because that's the most expensive part of this project, but they're banking the PBMCs so that if we need another set of another 1,000, that is available. So the exciting thing here is I think there's an opportunity globally for all the groups doing these type of studies to collaborate, and which they're doing already, so that we can share across nations the data that comes from these types of studies. And I do think that this is hopefully going to be a way to get at that heterogeneity. The other initiatives to try to understand heterogeneity are to develop better imaging markers. We all know from the field of multiple sclerosis that if you have a way to image the illness, you have a way to do really fast, effective phase 2 studies. We all have, or I personally have a little MS envy, and it would be phenomenal to have something like that in ALS, because I think it would really speed up drug development. So there is an international consortium that is looking at different imaging markers. This is just one example of a PET ligand called PBR28 that binds to activated glial cells. It's not specific to a particular M1 or M2, but it is a measure of neuroinflammation. And work led by Dr. Atassi at Mass General, he's now done this in probably over 50 people, and over time, he finds reproducibly this sign of inflammation in the brains of people with ALS, and that it is in a way related to the site of onset, with the bulbar patients having more of the activation in the brainstem, the limb onset more in the cortex. And so in the early days, it looks like this measure does not change over time. And we're now starting to do some studies in pre-symptomatic patients and people who carry, for example, the C9 gene or the SOD1 gene, to see how early this sign develops in a way similar to a lot of the efforts being done in Alzheimer's disease. So I think it's early days for imaging, but an important area and also an exciting one for people to go into. We are actually starting to incorporate that biomarker, that imaging biomarkers from early phase studies, particularly with a drug called Gludolast, which is a drug that works on neuroinflammation. We have an active study testing whether that decreases this PBR PET binding level in people on the Gludolast. So now I want to shift to what are we going to do with the clinical trials? And this is where I said that it's remarkably similar to the thoughts that Dr. Kim had, and I swear we didn't talk about it before. But I think this is where the field is going, and it's really about cohort enrichment. And you can do that in multiple ways. One is that is really going more on clinical phenotype. That's so if you don't have a biomarker or a way to do it biologically, can you do it with clinical information? And I think this is exactly what was done with Radicata, and it's what's being done with the study of Neurome, which I'll show you some data, which is a brainstorm stem cell trial, and another trial called Centaur, which is a combination of two drugs, Tugta and Busanol. So all three of these studies are targeting people who are early in the illness and rapid progressive. That could end up being a good model, but it also might not be a model. But it's a new approach of trying to cohort enrich by clinical phenotype. Another way, and one I think that is going to grow over time as we develop the tools to measure the biology of people, is to cohort enrich by biology. And there's already several examples of this. The retigabine trial is a drug that is targeting people with obvious measures of hyperexcitability. Because this is a drug that preclinically works on hyperexcitability. Ruxolitinib is a JAK kinase inhibitor that preclinically has been shown to decrease neuroinflammation, not only in ALS, but also in models of FTD and Alzheimer's. So a trial is just about to start, actually called a basket trial, which is where you can enroll people with different diseases but shared biology. And it's going to pick out people based on a biomarker of that neuroinflammation. So enriching by having a particular pathway identified. And then, of course, the most classic example for ALS is the targeted gene therapy trials for people with SOD1 or C9, and both with ASO approaches or viral delivery of ways to silence the genes. So I think this is very exciting for the field. It's new for the field to figure out how to do that, and I'm sure there'll be some successes and also some failures and lessons learned. And this really brings me to what I hope we'll be seeing in the next future, is that as a global community, we'll be meeting often and thinking about how we can be more innovative in these early phase trials together, using, again, some of these techniques that have now been well established in oncology. In particular, the idea that if you have 10 drugs that work on neuroinflammation, can you test them all in one trial? Share a placebo group, share the biomarkers that you're measuring, and actually see which one might be the most, the one with the highest potential to bring forward to phase 3. That type of approach has been done in Alzheimer's and brain tumors and other oncologies, but it requires a community that works together and that works with pharma and convinces people that this is better for the entire community. And I hope that this is something we'll be seeing in the next couple of years. Related to that is the idea of, can you have a master protocol, where once you can figure out what a good phase 2 design is, that instead of every time you have a new drug, you start over writing a protocol and giving permissions. Can you have an ongoing protocol that ethics board has approved, and every time you have a new drug, you amend the protocol? So again, shaving off probably 1 to 2 years off of each trial. I think the field needs to move there. We're at the point where we have enough ideas that it makes sense to kind of move in this direction. So I want to talk briefly about the first one, this idea of taking a homogeneous, more homogeneous clinical group. And of course, this was best example by Radicava. And I didn't put any of the data on Radicava because I figured that you knew it probably better than I know it, since it's been approved in Asia for quite some time. But really it does leave these 2 questions, if not some more questions. Was this a very good trial approach? Meaning that if you're going to do a short trial, taking people who progress rapidly or excluding people who won't change in that 6 months, is that just a good strategy for design so that you optimize your chance of seeing success? So that's possibility 2. Or is it there's actually something biologically different in these fast and early progressors, and Radicava, for some reason, was tackling that. And I'd say at this point, we don't know. I think the regulatory agencies have voted with number 2 and approved it for all forms of illness. But this is, I think, an important question for us to think of as we keep moving into more trials that are targeting these fast and progressive patients. So I want to just briefly talk about the brainstorm trial, and I know again that there's a talk later about the Neuronata trial. So I'll just say that there's a lot of interest and a lot of potential in using mesenchymal stem cells in people with ALS, and there's a lot of possible mechanisms of action. And I've been most involved in the trial with brainstorm, also known as Neuron. And last year we completed a phase 2 study. And this was a study really whose primary goal was safety. And people got one treatment with these mesenchymal stem cells that are grown in culture for a month to secrete high levels of different growth factors. And so people came in for about 3 to 4 months before. So we had a pre-slope treatment, and then they got one treatment, and then they got a spinal tap 2 weeks later. And we followed people for 6 months. There were 48 participants. It was a randomized placebo-controlled trial. It was a 3-to-1 randomization, so there weren't that many people who received placebo. The main outcome measure was achieved. This was very safe. There were no deaths or treatment-related SAEs, no dropouts related to SAEs. There were some side effects related to the lumbar puncture, but basically a very safe study. There were a lot of measures looking at efficacy, with the big caveat that this was a small study and single-dose treatment. So I'm going to walk through some of these because I think it led to how we decided to design the phase 3 trial, particularly targeting fast progressors. So there was a pre-specified subgroup, and this pre-specified group was picked because in the earlier phase 1 studies in Israel, they had a sense or some data suggesting a bigger response in faster progressors. So at the beginning of planning the phase 2 study, it was pre-specified to also look at these efficacy measures of people who were fast progressors. And that was defined as people who dropped more than or the same, equal to 2 points in that run-in period, so over that 3 months. So overall, there were the 48 people, and there were only a small subset of rapid progressors, 15 on the treated group, 6 on placebo. And there were a fair number of slow progressors. And again, in a small trial, having people who don't change during your trial period does make it very difficult to make and allow the claims around efficacy. So this is the overall data. So not using the lead-in period, just once you're treated in 2 groups, and using all the data. And you see on the left is all the data. The 2 groups really going the same. There's this little bump up in the treated group at 2 weeks. I say that fit clinically with what we were caring for patients. But this is the rapid progression group. So these are the people, the 15 on drug and 6 on placebo. And here we see the separation continue the whole time, getting an early bump up. Again, the patients only received one treatment at baseline. So then we looked at the data. Again, this was pre-specified in different ways, using something called a responder analysis. And this is the idea that not everybody in the trial might respond the same way to your drug. So can you define what response means? And we defined it in multiple ways. One was 100% improvement, meaning that you didn't progress. So if you were declining